Good evening to all of you. In this video lecture, we will be talking about a process called transcription. Transcription is basically a molecular mechanism in which RNA is being synthesized from DNA using DNA as template. DNA is double-stranded molecule, as you know. From this double-stranded DNA, a single-stranded RNA molecule is being synthesized. It may be mRNA or rRNA or tRNA or any other RNA like snRNA, scRNA, snORNA, etc. etc. All kind of RNA that are being synthesized from DNA, the process will be called transcription. So basically, it's a part of the central dogma which says that the information flows from DNA to RNA to protein. Information is being carried by the DNA in the form of genes. The genes are the information that actually determine the character of an organism. So the sequence of DNA present in the gene, in a DNA, that region of the DNA which synthesizes a functional RNA is called gene. So basically this gene is the information, and that information is being transferred to RNA via the process that is called transcription. So this is transcription. And this phenomena in which RNA to protein is being formed, this is called translation. Let us understand a little bit more about this. Say this is a double-stranded DNA, and it is a very long DNA. It may be very long DNA, maybe thousands and 10,000s or 1 lakh base pairs long DNA chromosome, in which there may be areas where genes may be present. In human chromosome, nearly 3% of the total genome is gene. In eukaryotic system, it is in E. coli, in prokaryotic system, the number of genes are quite more. The resolution of gene in the DNA is quite high. So suppose this is a very small part of the DNA, of the gene in fact, and if the sequence of this DNA is A, T, G, C, C, G, this is arbitrarily taken, 5 prime, 3 prime, a part of it. And this one, the opposite strand will be automatically T, A, C, G, G, C, 5 prime. So the RNA from this, suppose if this strand acts as a template, then the RNA that will be synthesized from this DNA will be, what will be the sequence of that RNA? It will be A, U, G, C, C, G, 3 prime. So you can see, if this is the part of the gene, say it is the total gene for the time being, let us understand the genes are actually not less than 150 base pairs long, or if comprises of minimum of 150 base pairs. So suppose it is a very long DNA, but for understanding, we are taking this 6 nucleotide long sequence to understand how the complementarity is being maintained. So if this is the template DNA, this synthesizes the RNA that is being synthesized is a complementary to this particular strand. So it will be automatically against T, A will come against A. T has to become, but you know that in case of RNA, T is being replaced by U. So in that way, this is the complementary sequence to this strand. So this is the template strand. This is the R template strand. And you can see that this sequence matches this sequence, except that U is being T here in the DNA. So the information that was in the DNA is being transferred to RNA now. And we know that these 3 nucleotide sequences, they specify a particular amino acid. So if a protein comprises of many amino acids, say methionine, serine, valine, so on and so forth, each of them will have 3 nucleotides in the RNA specified for them. So the information will finally being transferred from this to protein. The protein looks like anything like this, with an N-terminal and C-terminal end. It is a highly folded structure, three-dimensional structure, and it basically comprises the fundamental units of protein is amino acids. These amino acids, and if a protein comprises of 50 amino acid process, say for example, if there are 50 amino acids, the nucleotide sequence present in the DNA will be 150 nucleotides. There will be 150 bases in the RNA. In the DNA also there will be 150 base pairs for this RNA. So in the RNA that is being synthesized is exactly the copy of the DNA, except the T is being replaced by U. But the amino acids that are being synthesized, each of these 3 nucleotides, they specify 1 amino acid. So there will be, out of 150 base pairs, there can be only 50 amino acids. This is the relationship between these 3 molecules, and protein is the last molecule in the form of which the expression of particular character. There are 2 types of protein, structural and functional. Functional protein means the enzymes mainly, and the structural protein means the body that is being formed by the protein, that structural protein, the cells, the actin filaments, the microtubules, the cytoskeleton of the cell, and many things. Protein is part of our body is the structure, our hairs, our nails, and everything almost. So this is how the central dogma and the transcription process is about. So this is called central dogma. This flow of info, the concept of this flow of information from DNA to RNA to protein. This was given in 1958 by Francis Crick. This concept of central dogma. But later on, during 1970s, 1968 particularly, Baltimore and Temin, they discovered the new enzyme that is called reverse transcriptase. From viruses like Rous sarcoma virus, HIV, etc. These are called retroviruses because they can do the process, they can synthesize DNA from RNA. They contain RNA as their genome, and when they enter the cell, they convert this RNA into DNA, which gets integrated into the host chromosome. So this process, they can do this because they carry an enzyme called reverse transcriptase, which is the reverse phenomena. So when people came to know that the information can also flow from RNA to DNA, this central dogma concept was changed a little bit with this line, this arrow on the opposite side, telling that this is the reverse transcriptase process, that RNA can also be converted into DNA. So when you are drawing this flow chart of flow of information, you also should draw an arrow like this, which indicates that the replication of the DNA, which maintains the information. So basically, this is what is called central dogma. Now let us come to exactly the transcription process. We were talking about this process, this one, transcription. How does it take place? We have already made a video lecture on replication. There are 3 video lectures on that. You can go and see upon them. But this is video lecture specifically about transcription, so we will talk about this after central dogma. So the enzymes that actually convert this DNA to RNA, this enzyme is called DNA dependent RNA polymerase. Or you can, in the short form, you can also say RNA polymerase. These are also called transcriptase, as they do transcription. Okay. So, what is the structure of this enzyme, RNA polymerase? This enzyme comprises of 2 alpha subunits, one beta prime, and one omega. These 5 polypeptides, they make the core enzyme. And another polypeptide, another subunit, that is sigma 70 rather, it associates with the core enzyme to comprise the total enzyme that is called the holoenzyme. The core enzyme is capable of binding the DNA anywhere on the DNA. It can bind anywhere on the double-stranded DNA, but it cannot continue transcription in absence of the sigma subunit, because they cannot recognize the promoter sequence, which are the specific sequences present, consensus sequences present just upstream of the transcription start site. So suppose this is a gene, this is the gene part of this DNA molecule. So it will have a promoter region at this position. It will have an AT-rich sequence at this site. Because this AT-rich sequence, they will melt and they will create a transcription bubble like this, where transcription, when the new synthesis of new strand can start the process, can be started like this. Okay, so this is the transcription bubble, or transcription eye, you can say. So the presence of promoter indicates that there is a gene just downstream of it, and this promoter sequence is a consensus sequence. We will soon see what is the sequence of that promoter region in prokaryotic system. So suppose if this is a double-stranded DNA, and this is the transcription start site. Transcription start site is being written as +1 site. So the promoter region will be present at -10 base pairs upstream of this +1 site, and there will be another sequence at -35 base pair upstream. This is a consensus sequence. It comprises of a consensus sequence called TATA, T-A-T-A-A-T. So that is called TATA box. It is called TATA box, or it is also called Pribnow box. At -35 position, there is another consensus sequence that comprises of TTGACA. This is called recognition site. Now these two sequences, two sites, are very necessary in front of a gene. This is a gene which this sequence specifies that there is a gene at downstream of it. +1 is the start site of that gene. So an RNA polymerase will come and bind here. It will come and bind here. There are altogether 6 subunits of them. Let me take some of the color for alpha subunit. This is alpha subunit. So this is alpha, 2 alpha, 2 beta, no, not 2 beta, 1 beta, 1 beta prime. There is another, that is omega. This is omega. And since it has bound to the promoter region, that means the sigma is also associated with it. So this is sigma. So in holoenzyme of RNA polymerase, this total is the RNA polymerase structure. Okay. So in prokaryotes, there is only one type of RNA polymerase. So we were talking about the structure of RNA polymerase and the enzyme that synthesizes RNA from DNA in prokaryotic system. There is a DNA-dependent RNA polymerase. It is also called transcriptase. Transcriptase, not transcription, transcriptase over here. The process is called transcription. That's why the enzyme is called transcriptase. Now let us talk about a little bit about the eukaryotic RNA polymerase. In eukaryotic system, we find there are 3 types of RNA polymerases. RNA polymerase 1, 2, 3. There are 3 versions, 3 types of RNA polymerase altogether. The RNA polymerase 2 is basically responsible for mRNA synthesis, messenger RNA synthesis. And RNA polymerase 1 is responsible for RNA, ribosomal RNA synthesis. It in fact synthesizes not all type of RNA. It in fact synthesizes 5S rRNA, It synthesizes 18S rRNA and also 28S rRNA. It does not synthesize the 5.8S RNA. That is being synthesized by RNA polymerase 3, 5.8S RNA. And the main function of polymerase 3 is to synthesize tRNAs and also some small cytoplasmic RNAs. So these are different RNA polymerases engaged in different functions in eukaryotic system. They can be distinguished by if you treat them with alpha amanitin. Alpha amanitin is a toxin. It is a toxin. It is produced from a fungus, the poisonous mushroom that is being called. What is the name of that mushroom? Very colorful mushroom. It is called Amanita phalloides. This is an example of a poisonous mushroom. It contains alpha amanitin. That's why it is poisonous. As soon as it enters in our cells, in our body, in our cells, it inhibits the functions of RNA polymerase, particularly RNA polymerase 2. RNA polymerase 2 is more sensitive to this enzyme, this toxin, than RNA polymerase 3, and RNA polymerase 1 is in fact insensitive to alpha amanitin. So by treating them, these RNA polymerases with alpha amanitin, one can distinguish their functions. Their inhibition rates are different. So there are three types of RNA polymerases that can be proved by treating with alpha amanitin. Now let's talk about the process of transcription. How does it take place? So like any other polymerization process, there will be three steps: initiation, elongation, and termination. There are, in a particular long sequence of DNA in a chromosome, you can say chromosome length DNA, there are many, this is double-stranded DNA, say. There are many genes and their transcription start site. Suppose if this is the transcription start site, it will be designated as +1, and if this is the transcription termination site, so there is you can say from here to here, it's a gene. It is also called a cistron. So from here to here, it synthesizes RNA like this, an mRNA or tRNA or whatever it is. But let us see, this is mRNA. So basically, it's a transcription unit. From here to here, it is a transcription unit. It can also be called transcripton. Transcripton, and on will be attached with this as a suffix to this transcript. So transcripton means it is a transcription unit. A transcription unit comprises of a start site and it up to a termination site. The sequence of DNA present between a start site of a transcription and its termination site is a transcription unit or a transcripton. So there are many transcriptons in both prokaryotic and eukaryotic systems. As many genes are there, as many transcriptons are there. In case of prokaryotes, in fact, what happens, there are many genes, they are transcribed together in the form of operon. They act like an operon, operational unit. So in case of lac operon, there are three genes, Z, Y, and A, that specify for three different enzymes. The mRNA that is being formed is a single mRNA. So it is called polycistronic mRNA. Polycistronic mRNA. There are three parts of it. Z will synthesize beta galactosidase enzyme, Y will synthesize lac permease, and A will synthesize transacetylase. We will talk about lac permease later on. And it has a promoter region in this position, promoter, and there is an operator region also. From here, +1 site starts. And there is the regulation of lack of iron, that is a different issue, we will see it later, but we were talking about the transcription unit. So in case of prokaryotes, you can say that this the transcription starts from here, it ends here, so it is the whole polycystone is a transcription unit in this case. So the transcription occurs in 3 steps: initiation, elongation, and termination. Initiation means the RNA polymerase will first bind to a particular site in the DNA. Say the previous diagram will be sufficient. Let us draw the RNA polymerase in a single subunit like this. This represents the whole RNA polymerase, this particular structure, rounded structure. And say this is the +1 site. So the first thing that will happen in initiation will be the binding of the, let us say this is the core enzyme and this is a sigma subunit associated with it. This is sigma subunit associated with it. So it recognizes the promoter region at -10 and -25 base pair upstream of +1 site. At that position, this RNA polymerase binds with the help of this sigma subunit. So what will happen next is that this part of the RNA, of the DNA rather, it is DNA, this part of the DNA will what is the color of this? This part of the DNA, no, this one, no, I'm a little bit colorblind, this one, I don't know whether this is matching. So this part of the DNA will get melted because it is AT rich also. You know that the promoter region is starter box, so there is AT rich. It will melt like this. It has to be melted because double-stranded DNA, they are twined around each other in a very tight manner, and new DNA, RNA synthesis or even DNA synthesis in case of replication cannot occur until unless that part is being destabilized. So basically the double strand, the hydrogen bond between them is broken, and the polymerase sits here like this. Again the color is being changed. Okay, you consider this as the previous one. So say this is the +1 site where the transcription will start, and this is where the Tata works and the recognition site also -10, -30. So the initiation the initiation means when this melting of the double-stranded DNA first and bringing of 2 nucleotides at this position at the start site. Say if the start site constitute of a T and a A, then the RNA, the 2 nucleotides that will be brought here will be, of course, that will be A and DU. Okay, and a covalent bond between them will be created. That covalent bond is the phosphodiester bond. So a covalent bond between them will be created by this RNA polymerase. So this completes the initiation process. As soon as these 2 nucleotides are being connected by this covalent bond, when the phosphodiester bond is being formed, the process of initiation can be said to be ended, and from here the elongation process will begin, because the molecule will now get elongated from you. So in elongation, say this transcription bubble, this is called transcription bubble, the melted area, or I, transcription I. This will gradually move forward. The helix in this region will open, and this will gradually move forward. So suppose it has reached to a little bit to this position where about 8 to 9 base pair it has shifted towards downstream. So new RNA has been synthesized, say, this length, about 8 to 9 nucleotides long. When this RNA, the 5' end will be a triphosphate like this. So when this RNA is about 8 to 9 nucleotides long, the sigma subunit, this is the RNA polymerase. Now the sigma subunit, it lifts the RNA polymerase. Sigma subunit is released. It lifts the RNA polymerase, then it is recycled. The next round of transcription will again start here at the +1 site, and this sigma subunit will again bind with the coenzyme to recognize the promoter sequence. So elongation process is going on as soon as this RNA is 8 to 9 nucleotide long. This is called the nascent RNA. The sigma subunit is released and it is recycled. In place of sigma subunit, another protein that is called NOS-G that comes and binds with the RNA polymerase, core RNA polymerase. There is no need of sigma subunit anymore because that was needed for recognizing the promoter sequence, and that job has been done. So after that, the polymerase can move itself forward, but NOS-G is necessary. The function of NOS-G is very interesting. It actually helps to understand the pause site and the stop site. It helps to distinguish between a pause site and a stop site. What is this pause site and stop site is? In the DNA, there are many sequences which are complementary to each other. These regions are maybe palindromic sequences at various positions. These palindromes, when RNA is being synthesized, that RNA forms at different, when they reach these positions, the transcription reaches these positions, the complementary region, they hybridize among themselves and they form a hairpin loop-like structure like this at various positions. The RNA forms like this, not the DNA. So if this is a pause site, and if this structure is followed immediately by U stretch of U about 6 to 8 nucleotides long, then that will act as a stop site. This is a termination site, but this is not a termination site. These are pause sites. So how a system will distinguish between these two? This is NOS-G, which helps to overread these pause sites and to recognize the actual stop site, and at the stop site the transcription is terminated. So this is how the NOS-G helps to understand, and it increases the processability of the RNA polymerase. Then after this elongation is going on until the termination site is reached. As we mentioned, the NOS-G helps to proceed further the all sites, and gradually the termination site comes now. Termination site is being say here is the termination site. RNA termination site. Transcription termination occurs. The termination sites are of two types. One is called row dependent. Another is row independent. The previous stop site that we mentioned, that a hairpin loop-like structure followed by a stretch of U in RNA. In DNA, what will be there? Stretch of A will be there. Against A, this U will be transcribed in the RNA. If this structure comes, the transcription will automatically get stopped. This is row independent method. But in case of row dependent method, the DNA contains a particular sequence at this position. These are called RUT, row utilization sequence. And this, so the termination site in transcription comprises of two types of sequences in the RNA. One has this poly-U site following a hairpin loop-like structure. This is hairpin loop-like secondary structure that is being followed by a stretch of U. In the DNA, the sequence will be in place of U, it will be A, continuous A up to 6 to 8 bases long, and so that will be transcribed as U in the RNA. And if this is one type, this is row independent type. Another is row dependent type that contains a sequence, a consensus sequence here. It does not contain this hairpin and this kind of stretch of U. It is different. It contains a sequence called RUT, row utilization sequence. So basically it is a consensus sequence. So how the termination will take place? Termination will be again of two types on this basis, row dependent, row independent. So let us first see. Let us divide it into two types. This is, let us first discuss row independent process. In most books you will get this process, row independent. Row independent means there will be no need of any row protein, RH row. This is termed as row. There will be no need of any protein, that is called row protein, and termination will automatically occur when this particular, so this is the termination site we are talking about. This is the termination region. When the RNA has reached, RNA polymerase has reached this area, and this particular site contains a stretch of A. In the RNA, automatically there will be a stretch of U over here, and that will be the upstream region of that will have a secondary structure like this, the hairpin loop-like structure, and this is the long RNA that has already been formed. 3-phosphate molecule here in the 5' end. So when this particular structure is, this is called attenuator site, this particular hairpin loop-like structure followed by U is an attenuator. So when this particular structure is raised during transcription, the RNA polymerase automatically is released. It gets off. It cannot hold this part, and the RNA and DNA hybrid easily melts away, and they get separated. The RNA gets separated like this. It forms a hairpin loop like this. This is the 3' end. This is the 5' end. And the double-stranded DNA, it reforms it. It rehybridizes. It again forms a double helix, and this RNA polymerase is being released from the system. RNA polymerase is released. This is RNA polymerase. This is how row-independent termination takes place. Row-dependent termination requires the presence of RUT sequence in the DNA and in turn in the RNA also. So this is the stop site, say. And it contains a RUT sequence somewhere here. So when transcription is taking place in the transcript, the RUT sequences, this is the RUT sequence region, and this is the RNA, the mature RNA that is being formed. This is bacterial system. So when this RNA is being formed in bacteria, what will happen? Ribosomes will sit here and will start synthesizing protein. Because in prokaryotes there is no nuclear membrane, and so the transcription and translation, both these processes are coupled. They take place at a time. As soon as a little bit of transcript is being formed, RNA has been formed, the ribosome get access and it binds. It recognizes the initiation site, signed on RNA sequence, etc., and starts protein synthesis. So whatever be the case, that is not important here. Just I gave a little bit of information. Let me remove these ribosomes. So let's say this is the RUT sequence site where this transcript has also got this RUT sequence here. So RUT sequence has a row utilization sequence, it is termed. It is a consensus sequence. It is capable of binding row proteins. Row proteins are basically a hexameric structure. It takes a hexameric structure, 6 subunit of row protein, they assemble together, and the RNA, it wraps around this. In fact, it wraps around this these 6 row subunits, row proteins. These are row proteins, and this particular the region with which this interaction occurs is the RUT region. And at the same time, when this is going on, Then another NUS protein called NUS-A. So this is NUS-A. It comes and associates with the RNA polymerase and this row proteins. This is RNA polymerase. And when these three things come together, the row protein, the hexamer of row, the NUS-A, and this RNA polymerase at this site, what happens? ATP hydrolysis takes place, and using the energy of this hydrolysis process, the RNA DNA hybrid is melted away, and the RNA is removed. The RNA is released from this. Let me write like this. This is the RNA. This is the RUT site. Now, this is 3' end. This is 5' end. The 5' end will have 3 phosphate molecules like this. Okay, and the DNA will also get rehybridized. It will come to its native form like this. It will again form the double helix, and the RNA polymerase, these row proteins, all of them will get released from the system. The row protein, they will be released like this. They will not be in a hexameric form now. They will be separated like this. And NUS-A will also get released. The RNA polymerase will also get released. So transcription termination takes place like this. So this is how the initiation, elongation, and termination of transcription process occurs taken together. Let me rectify a small error that has occurred a little while ago while discussing the types of eukaryotic RNA polymerase. A little bit information. This one, let me see whether it is right or not. Okay, this one will be that's what I was thinking. This will be 5.8s rRNA, and this will be 5s rRNA. This error is quite useful, quite common. You might do this error. The RNA polymerase 1, it synthesizes actually it synthesizes a 35s rRNA at a stretch, and that is later on processed into these 3 types of RNAs. Before that, it is actually 45s rRNA. Then it is clipped to 35s, and then it forms these different types of RNA. RNA polymerase 3, it transcribes the 5s rRNA as well as the tRNA and small cytoplasmic RNA. With RNA polymerase 2 synthesizes mRNA, plus it also synthesizes snRNA, SNOPs. Not the SNOPs, not the protein, it synthesizes the RNA part of that protein, snRNA, small nuclear RNA. So this is the process of transcription in prokaryotes, and in next video lecture we will be talking about how RNA processing, post-transcriptional processing takes place in case of eukaryotic system. In prokaryotic system there is no post-transcription because as a little bit ago I told that that transcription and translation process are coupled. In prokaryotes, what happens, this is the cell, suppose it's a bacterial cell, and this is the chromosome, suppose this is the open double-stranded circular chromosome, and transcription is going on. So this is the transcript that is being formed here. There is no nuclear membrane, ribosomes are there here and there all along the cytoplasm. So they will automatically come and they will sit here as soon as the RNA is being synthesized, and they will start synthesizing proteins also. So what is this? This is the transcription process. This was transcription. This is RNA, and this is protein. So protein synthesis and RNA synthesis both go side by side in prokaryotes. But in eukaryotes, what happens? In eukaryotes, it is different. In eukaryotes, you get a nuclear membrane. The cell has a, so this is the cell. It has a nuclear membrane like this, isn't it? You can make it like this also, if you like. Okay. So this is a nuclear envelope. Inside the nuclear envelope, the DNA is present, the chromosome is present, and transcription is taking place inside this nucleus. RNA is being formed inside the nucleus. Ribosome resides in the cytoplasm, so the ribosome cannot get access, it cannot enter in the nucleus. Rather, the RNA has to come out. So the RNA, when it comes out, these ribosomes get access to bind here and protein synthesis starts. So here you can see that the protein synthesis, the translation, this part, translation, and this is transcription. Both are temporally and spatially separated by the nuclear membrane. And this is required because the transcripts that are formed in case of eukaryotes are not the mature transcript, they are called hnRNA, as they contain the non-coding sequences also. hnRNA, heterogeneous nuclear RNA. Heterogeneous nuclear RNA comprises of both exons and introns, both. Exon, intron. You know, exons are the coding regions, intron are the non-coding regions. So first, from the DNA, when transcription takes place, the entire DNA sequence is being transcribed into an RNA called hnRNA. This is premature RNA. Later on, these intron parts, the non-coding regions, they are being removed by a process called splicing. And what ends up is only these exon regions, they are present in the mature RNA. mRNA contains only the exon part, not the intron part. The intron part are removed through splicing. These kind of modifications are called post-transcriptional modification. Along with this, some end modification occurs, like in the 5' end, a guanyl molecule is added, and that molecule, and the 7' position of that molecule is methylated also. This is called 5' cap. And at the 3' end, a stretch of A, about 150 to 250 nucleotide long A residues are being added. This is called polyadenylation. These are end modifications. All these processes, end modification and as well as the splicing, are together called post-transcriptional changes or post-transcriptional modification. Therefore, in eukaryotes, for this post-transcriptional modification to take place, this nuclear membrane is being provided. Within the nuclei, the post-transcriptional modification like the splicing and the end modifications occur. When the processed RNA is brought outside the nuclei, the translation occurs. Therefore, in eukaryotes, we find transcription and translation in separate places, that separate time-wise also, as well as space-wise also. So that is what the significance of nuclear membrane is all about, why this nuclear membrane is present in eukaryotic system and it is absent in prokaryotic system, because prokaryotic genes are not split gene. They are continuous. There is no exon intron kind of thing in prokaryotes. In fact, there are many genes which are present one after another, as we talked about the lac operon, and they constitute an operon sort of thing. This is because of the space problem. In prokaryotic system, there is a single chromosome with many genes. Say, for example, in human beings, there are 20 to 21,000 genes in 23 pairs of chromosomes, which are very, very large chromosomes, in fact. Whereas in case of an E. coli, like bacterium, there is a single chromosome and it contains about 4,000 genes. So, 4,000 genes in a single chromosome will definitely be very compactly arranged. And that's why they prefer operon kind of operational units. Genes are being arranged side by side, controlled by a single regulator promoter in operator system. Thank you very much for your patient listening. And at the end of the video, I would request you to subscribe this channel and like this channel more and more. Also click the bell button so that you can get new information about new videos. Thanks a lot.